it's difficult to say, when we're going to solve things, because very often what you hear is that, but the first person that will live to 150 is born already, or to live to 200 years, etc. And we can never say, it's very difficult to predict what is around the corner, like in the future, what will we solve in the future, given these new methods and all of that data that we're producing. But today, the things that we know today in terms of intervention, supplements, drugs, lifestyle changes, etc., we will not solve aging by just the knowledge that we have today. So I think in order to live longer than maximum lifespan today, we need to solve some of the complexities of cellular aging.
Today we're joined by Dr. Sara Hägg from Karolinska Institutet. She leads the molecular epidemiology of aging group and works at the frontier of what might be one of the most talked about tools in longevity science, biological age clocks. These models promise to tell us not how long we've lived, but how fast we're aging, a kind of molecular speedometer that could one day guide prevention, clinical trials, and maybe even the way medicine is practiced. But how accurate are they really? What do they measure, and are we anywhere close to using them in the clinic? That's what we'll explore today with one of the leading researchers in the field. Welcome, Sara.
Thank you very much.
How did you end up doing what you do today?
I guess as a researcher, you follow your interests. So it's more of different circumstances that makes you end up somewhere. So I can't say that I had a typical strategy, but I started to work. I did a PhD with more cardiovascular focus, and then I started as a postdoc to work on other age-related diseases such as dementia and cognitive decline. So the commonality was aging. So eventually, with my interest in biomarkers and the biological background that I have as well, started this research line into the biomarkers of aging and that interest.
We have several guests who have these have had this epiphany when they were young, that they ask themselves, why are we going to die? And that's horrific. Was there something of that involved for you? Or was it more that you started working on adjacent areas and then ended up in aging?
Yeah, I mean, of course, I find aging fascinating and that also drives my interest. But I can't say that I really have, been wanting to do this for as long as I remember. But going back, I can see that I wrote some essay in high school on Alzheimer's disease. So apparently there was an interest earlier that I kind of forgot about it. But an interest, but not really something that I thought I would really pursue.
And you working from Sweden, it has always seemed to me that we're not at the forefront of this longevity science, geroscience paradigm in Sweden. Do you agree with that? Is it changing? How do you view that?
Yes, I do agree. I think that we are a bit slow in Sweden sometimes to pick up new trends, etc. So that's also why we are perhaps a little bit behind other countries. But I think also there is a growing interest now. So even traditional medicine and people working in the clinics, etc., starting to recognize this. But of course, it has also been a challenge that people don't really feel that aging is something that we should do. Put our focus on.
Is that something you encounter at Karolinska?
Yes, sometimes, but I think the last few years have been a shift. So I can see it also here. I'm obviously internationally with lots of conferences and invitations to talk in those places, but also here in Sweden. So I get more invitations to, of course, a lot of public outreach events, but also to conferences that are targeted towards clinicians. And I try to, whenever I get those invitations or to do a seminar or something, I always try to do it because I think it's very important to get the interest in the people working in clinics today as well.
I guess still today the paradigm is to research into how to manage aging, not so much in how to retard it.
Yeah, I mean, absolutely. We have a lot of research in geriatrics and care of the elderly, etc., and not so much into the biology of aging and the mechanisms behind it, and trying to understand those paths. But there are a few groups at Karolinska that does some experimental work, preclinical work on aging. So it's not a big thing, and it's not any. We don't have any specific grants for biology of aging in Sweden, but I can sense that there is a growing interest, at least, which is good.
When you say it's not a big thing, how many people are we talking about?
What do you mean there? That how many people?
Well, how many people are working on trying to solve aging in some way?
In Sweden, or?
Yeah, in Sweden, yeah.
Oh, it's hard to say. If we go really to the mechanistic part, so not into gerontology or geriatrics, but more into the mechanistic, yeah, maybe it's still a handful of groups in Sweden. So we more or less know of each other. It's not that many groups.
100 people?
Altogether, yeah, maybe not that many. I would say a bit fewer, yeah.
We pride ourselves of being a great life science nation, which we are in other respects. But
the reason I was pressing on a number is that we often hear, Peter and I, that it's such a big thing now, research into longevity. And so I think it's important to put a number. It's quite relative, and this is relatively small.
No, that's true. So the relative number of people working on aging mechanisms and aging biology in Sweden is very few compared to other areas. But what we do is that we try to combine efforts across the Nordic countries as well. So I just came back yesterday from Iceland, where we had a Nordic Aging Society meeting. So we try to have them now annually to really also get the growing interest and to get increased collaboration between the Nordic countries, because of course we face a lot of the same issues across our countries.
And one of the things that Sweden do have is some pretty special data, both from twin studies and other registries. And you have chosen to do population scale work when you're looking at biomarkers of aging. So tell us about that special repository of data that we have.
So this is special to Sweden, but also to other Nordic countries again, that we have registries, and that means that we keep track of individuals using our personal identification number. And we have that across a lot of registers. And this is a good resource for doing research. We can link different registers together, such as health data registers. So we can really track down the total population. So not just single down on different hospitals, etc. We have national-wide registries that we can link together. So that is fantastic. So traditionally, we're very strong in epidemiology because of these registers. But we can also link them to other cohorts and other data sources. So they can be used as something that we put on top of other information that we have, which is really good. And also the Swedish Twin Registry is the largest twin registry in the world, but there are many twin registries as well. But here again, we can link it to different national registers as well. So it's a very rich source in many ways.
And you also use the UK Biobank. What is that for someone who doesn't know about it?
Right. So the UK Biobank is also, I would say it's a population-based cohort collection. They call it a biobank, which is a bit confusing because it's not what we call a biobank in Sweden. A biobank in Sweden is just a collection of samples. But in the UK, it's a population-based cohort of 500,000 individuals. So they have collected data on these people and then also follow them in registers. Maybe not as many registers as we have in Sweden, but they do add on that type of data as well. And then they collect information on different biomarkers and such as genetics and proteins, etc., and health-related questions.
Okay, So now we have a clear understanding of the data that you're mining, but maybe we should explain what epigenetic age or biological age is for someone who doesn't know that, and how that field really started.
Yes, so when we talk about biological age, it's really a relative measure of the health, the physiological or functional health status of your body. So if you would compare yourself to other individuals that are chronologically the same age as you can say something about your relative health. So are you healthier than your colleague that the same age, for example, or vice versa? And you can estimate these type of health measures using lots of different biomarkers that are connected to basic mechanisms of aging, so that we know that they change with age. And on a population-based scale, we also know then that all sorts of these types of assessment, biological age clocks, we call them, is also related to the risk of different health outcomes, such as future diseases or mortality. And the field of epigenetic clocks, which is then the biological age assessment using epigenetic data. So this is one type of molecular data that you can use. So it's an assessment of DNA methylation from the whole genome perspective. So it's also something that is added on top of your DNA molecule, but it regulates how the DNA is expressed. So for example, we have the same DNA in all of our cells, but we use it differently. So a hair cell is different from a cardiomyocyte or from a skeletal muscle cell. And the epigenome is used to regulate this differences, you can say. And it turns out that about 10-15 years ago now, the first studies came out that linked epigenetic mechanisms to aging. Because the way that we regulate these differences to manage our gene expression in different cells is also changing while we are aging. So it's changing across the life course. So this is why that if you measure DNA methylation, which is one type of epigenetic regulation, you can also use that information to build an epigenetic aging clock, which is very accurate then in terms of assessment for different health risks, etc.
We mentioned methylation quite a lot. What is methylation really?
So methylation, it's a chemical modification. So it's a methyl group that is added, that can be added or taken out from the DNA molecule. So it's added to the cytosine-guanine base pair. So it's particularly for that type of base pair in our DNA molecule. There are also other types of epigenetic marks. So this is one type of chemical modification. So we call it chemical modification because in theory, you can add them and you can remove them. This is really the way that it's either blocking or enhancing expression of certain genes. So it's like a switch, like you turn on and off your lamp. It's kind of the same thing it's an analogy to turning on and off a gene, basically.
Yeah. What happens with thymine and adenine then? Aren't those expressed differently as well?
So the methyl group it only adds to the cytosine-guanine pair.
Yeah.
But there are other epigenetic mechanisms, and
Right.
I'm sure that there are lots of regulatory processes that we don't even know exist today. So there are different chemical groups, acetylation, lactation, etc., that can also be added to other molecules, the histones, and that the DNA molecule wraps around, etc. So there are many of these regulatory processes, and I'm sure that they are all connected in different ways as well. And it's really super complex, and I only think that we're starting to scratch the surface to understand these mechanisms. There are also short RNA sequences that can be considered to be epigenetic regulatory mechanisms, and those are huge. There are very, very many different types of short RNA sequences as well.
How come methylation groups has been so prominent then?
Yeah, that's a good question, because obviously we know that there are many mechanisms. So very often what happens is that the knowledge that we have is the growth of technological readiness. So whenever we have a method where we can measure things accurately and we can do it large scale, high throughput in large numbers, this is when we start to see a big growth of knowledge within a field. And this is exactly what happened with the DNA methylation array. So it's basically a ship, a small ship where you put all your samples on, and you can then easy, quite cheap, do it high throughput to measure this in samples from humans or animals or other species. And I think this is also a natural continuation of the growth that we saw in the human genetics field, where we have these different arrays to measure genetic variants as well. So this is kind of the same technology that is developed, which makes us basically it makes it possible to measure these things at large scale, so that we have the data to do these type of studies. So that is why it has been such a growth in this area.
So that was why it paved the way for the clocks.
Yeah, exactly.
Yeah. So tell us a little bit about how that started. Was it with Steve Horvath's foundational work there?
Yeah, so it was the work by Steve Horvath. He published a paper in 2013 in Genome Biology, I think it was. So he was the single author on that paper. So he used a lot of publicly available data from the first types of these DNA methylation arrays that were available from human data. And then he actually worked on a different project. He had a different study aim. But accidentally, he found that there was such a strong association with age in all of the data sets that he looked at. And about the same time, there were actually other groups as well that had found this. And that started this whole idea that you can identify sites. We call them CPG sites because this is where the methylation happens across the genome. And you can identify different sites that then are changed across the life course like that.
And wasn't it the case that his results were so striking that publications hesitated to publish his study?
Yeah, I'm sure. I don't know the full story behind it, but
I think it was,
I'm sure.
It was too accurate, his clock.
Yeah, exactly. It's too good to be true.
But okay, so that was 12, almost 13 years ago now. So now there are many different types of clocks. There's been a huge development on that side. Can you? I think we're going to spend some time on those that are most prominent. But could you just briefly explain what the big trends are here and how that has developed over the years?
Right. This new methods that came out, a lot pioneered by Steve Horvath, made it possible for many more groups and researchers to develop these clocks or to apply them to their own data, as more and more groups had these DNA methylation data available. And what Steve Horvath also did that was quite smart, I think, is that he made, so he's a statistician by training, so he made also codes for developing the clocks that he had. He trained on data, and then he published that, and then he made the clocks that he developed available to the field. And he made software for people to use them, to apply them to their own data. And this was a very smart thing to do. So now we had the data and we had the method to do it. And it was simple and people could use it on their own data. So this really started this growth. But of course, at the same time, other types of data became available. And also one quite popular method is also using what we call a pheno-age clock. And this is normally a combination of different clinical biomarkers. So it can be different sets of clinical biomarkers, but many of the biomarkers we normally measure if you do a health check, for example, they change as we age. So blood pressure, blood glucose, blood lipids, etc. And you can combine these in different ways to create something that we call the phenoage clock as well. So there are methods for that, and those methods have been partly developed maybe even 20 years ago. So, or there were also things happening before that, but they started to become available more and more now as well. And on top of that, what we see now is much more large-scale omics data. So we have metabolomics and proteomics, for example, and RNA sequencing data. And all of these type of omics data sets, you can basically apply the same type of strategy as for the DNA methylation data and train models to predict age using that type of data, and then you build your own aging clock based on that data type. So it's been a huge growth, I would say, in the field to apply this also to other data sources.
Omics, for someone who doesn't know what that is.
Right. So omics, I mean, the word means totality or of some sort. So whenever you do like epigenomics, it means that we're studying the epigenome, we're measuring epigenetic marks across the whole genome. So we wanted to measure the totality, all of it. I mean, it's obviously we're not measuring everything, but that's the expression. So it's the same thing with transcriptomics, which from RNA sequencing data, for example, you measure everything, all the genes that we have, or proteins, we try to measure as many proteins as possible. So it's really a growth in technology as well that enables us to measure many things, many molecules at large scale. So trying to capture as much as possible of the totality, this different omics layers then.
Yeah. And then there are something called PC clocks, I think.
Yeah.
I don't know if you call them PC clocks or if you call them something else.
No, we do, but I think the method is mostly developed still for epigenetic clocks. I'm not sure if anyone has applied that principle to other types of data or clocks. But what it means is that, so PC clock, PC stands for principal components, and that is a computational method to reduce dimensionality. So that means that if you have a lot of data, you're measuring the totality again. Maybe you need to zoom down or to somehow get it down into something that is meaningful, or you want to zoom into the things that are important for aging, for example. Then, of course, you can use these type of data reduction methods. So principal component is one such method. And we applied that to epigenetic data and the specific epigenetic clocks because we wanted to try to separate even within the clocks, and they are trained to predict age, but there are a lot of noise. So there is much variability and a lot of uncertainty in these methods as well. So we try to really use that method to separate, okay, what are the big chunks that are really important for aging, and what are other things that are maybe more related to noise or technical variability, etc. So it's a way to try to separate that, because what we need is to have more accuracy in these types of clocks. And I think we will maybe talk more about that, but that was the way in this paper that was published a few years ago that we contributed to try to improve the epigenetic clocks that we had.
Yeah, I mean, there's a whole Biomarkers of Aging consortium. It's one of the most key areas in aging biology, I would say, because and most people, I think, or many people think of these clocks as a way to measure their own biological age or epigenetic age, but that's not why you're working on it, I presume. You are working on it because we need this to solve what, Sarah?
I mean, it's one piece to the puzzle. And of course, what we say is that in all different fields, there are biomarkers. You need markers to detect what is going on. And that's also true for aging. We need markers so that we can study the aging process. And I'm interested in human aging, so I want to measure this in humans. But to do that, we need to do a lot of studies, because today, we don't have any gold standard biomarker of aging. We don't know which one is really the best one. We don't know what the best method is and how to really reduce all of this noise levels that we detect using different types of data. So this is really what the consortium of biomarkers of aging have been trying to engage in, to try to get closer to this. How can we set up guidelines, strategies, standardization protocols, so that we can eventually get clinically validated biomarkers of aging? And if we have that, then we can start to use that at a broader scale. We can do clinical trials with them and better translational studies, and perhaps also use them in preventive healthcare treatments eventually. But we need to do all of these steps beforehand. I mean, that's what they've done in other fields, cardiovascular field, whatever, to standardize and have these type of markers. So we're trying to set that ground now.
Yeah, we need it has to be like a really good crystal ball. It has to tell us something we can depend on for the future, right? Otherwise, we can't really rely on it for clinical trials. We can't use it in medicine because that might pose a danger to the patient and so on. Is that correct?
Well, I mean, to start with, we have to do the studies in research, and this is where I do a lot of the work. That's where we start, basically. But taking this results from research and apply it to the clinic, then we have to have many more steps, of course, because now we show that they work on a population level, they work in research studies. But in order to get them to work individually and in a meaningful way so that we can use them in clinic, they need to be much more accurate. And we also need to understand not just the accuracy, but we also need to understand whether or not they reflect changes. So if we modify something with an intervention or a lifestyle change or something, and we can measure that on a biomarker scale, does it also mean that there is a true improvement of health, or is this just something that is correlated, etc.? So we need to also understand more about the basic pathways and the causal pathways, really, which I think we're still lacking quite some work here.
Some of the fine-grained mechanisms here, actually, the methylation and the methyl groups. What is it about them that signifies that they belong to someone who is chronologically older versus younger? How can you see that?
Let's say that you have, for a certain gene, you have an upstream region where you can see a lot of methylation. And if the upstream region is methylated, there's a lot of methyl groups that has been added. It means then that the transcription cannot start. So the enzymes that are responsible for reading the gene, they cannot bind to the DNA molecule. So they cannot start to read the gene. So that's why it's kind of a switch. And what we can see is that many of these upstream regulatory parts of different genes, they get more and more methylation as we age. So it's what we see is that there is some sort of silencing of genes as we age,
too much silencing.
That was a little bit of the explanation.
Well, I mean, so it's not that the process itself is different. It's that you can see it where it shouldn't be, or there's activity where it shouldn't be activity. Is that correct?
Well, we can see that there is like an accumulation of methylation as we age in these regulatory sites. And of course, that methylation also is then influenced by other factors. So for example, genes that are important to express if you smoke, maybe you need to have certain types of genes to be expressed in your body so that you can handle the smoke that comes into your lungs. And because of that, you activate those genes, and then that will leave marks from methylation on those genes. So this is why we can also build clocks from epigenetic data, not only on aging, but we can build a smoking clock. We can build a clock that tells us how much we've been drinking coffee or alcohol, etc. So it's actually leaving marks on our DNA molecule from many different things happening across life.
So to take an example, a skin cell, for example, the accumulation of methyl groups where they shouldn't be is, for example, preventing the DNA to be translated into these cells by inhibiting where it shouldn't be inhibiting. And so the cell becomes less flexible, for example.
Yeah, but I'm not sure that you can say that it shouldn't be methylated, because everything, what we say in science is that everything makes sense in the light of evolution. So, of course, there's a reason why this is happening, why we have these epigenetic mechanisms. So there must be a reason why we eventually turn off different genes with age. And I'm not sure that we understand the full picture yet. We don't really know why this is. And if it's so simple that you can just remove these groups and then we will go back in time. I mean, those type of experiments are ongoing, that we're trying to reprogram the epigenome. But it's not easy, I would say.
But it is the one of the core explanations for example, of course, skin aging.
Yeah, I mean, at least it's maybe a correlated marker. We don't know if you go out in the sun and you expose your skin to a lot of sun, we know that you also age your skin faster, for example. And that process may be because of this, the sun expose our skin, which starts some sort of epigenetic reprogramming or remodulation that then affects our genes, etc. So it's kind of in statistics we would say it's a mediator. So it's something that is, in the path, leading from one thing to another. But to say that it's completely causal, we don't really know, actually.
What types of clocks are you working on now, and what interests you most in the field?
We work a lot now with the proteomic data. And we also have new metabolomics data in our some of our studies. And with the proteomic data, maybe we'll come back to that, but there is a big growth now in studies where we try to look at organ-specific aging based on proteomic data. So we can talk more about that. I think that's very interesting, actually.
Yeah, please do. I mean, maybe first of all, it's interesting because we've been talking about epigenetics now and how methyl groups change the expression of a gene, silencing it and so on. But proteomics data is kind of downstream of that, right?
Yeah.
So why is that potentially interesting? Exactly.
So it's downstream. I mean, first we have our genes, and then we have the epigenome that is controlling how the genes are expressed, and then we have the transcribed RNA, etc., from the genes, and then later we have the translation to proteins and perhaps also metabolites. And so it's like you say, it's proteins are more downstream, so we would say they're closer to the clinical phenotypes and symptoms. So in that sense, it may be a better marker if you want to study associations with certain diseases, because it's later in this process compared to the epigenetic marks. So I think proteins are for that reason it's really interesting, and also they are more in the circulation. If you plasma and serum from blood, for example, you can capture proteins that have been secreted from different organs in the body. So this is why it's also the DNA methylation data from whole blood. Then that's basically from our leukocytes, our immune cells, whereas the proteins can come from all sorts of different tissues and organs and systems.
So then you can make organ clocks.
Exactly. So we can look at the proteins that are secreted from the liver, for example, and then we can build a clock based on the liver proteins secreted from the liver just from taking a blood sample. So we can do that for a number of organs, and it also works in the way that, for example, if we create a liver aging clock based on those proteins, it will also predict liver disease higher than if we have another type of clock created. So we have this more specificity then in the terms of picking up the proteins from different places.
What do we know about organ aging at this point? Because isn't it true that different organs age at different rates, and that's also a reason why it would be important to have these more specific clocks?
Exactly. So you can find that within an individual, you can have different aging rates at different organs or systems. Obviously, a person that has a very aged heart is also more likely to have aged arteries and aged kidney, for example. So there is a correlation like that, but there's also this separation that you can actually see different specific aging parts in the body. So again, like I said, that it's then been shown in some studies that if you have an increased or accelerated aging of your kidney, that also means that you have a higher risk to develop kidney failure. So there is this specific association as well. So I would say that the organ clocks are complementary to the general aging clocks that we've seen mostly from the epigenetic clocks, because you also want this general aging clock of the whole body, but maybe it's like, I would say that they're complementary because you also want specificity. I think that the organ-specific clocks from proteomics, it's more closely related to what we have and use today in clinics. If we we go to the GP and we do a health checkup and you do the clinical biomarker tests, you would test your liver enzymes. So that is also, which would be an indicator of how your liver is doing.
Yeah.
So this is similar to that, but it's in a little bit different way. And then if we can scale it up to many different organs and systems, that could have immediate clinical implications, I would say, if we could get it to be accurate enough.
Do you think it's likely that we at some point will have some kind of that the standard will be some kind of super clock that incorporates a lot of different clocks and you kind of triage them to get an accurate measure of what it is you want to find out?
Yeah, I mean, with this, you can, if this is growing, I think you can get both specificity for different parts of your body, but also the generalized estimate of how you're doing in general. So that is a strength, of course, if we can use both perspectives.
What are the big bottlenecks now? What are the biggest challenges that you face?
Yeah, that's a very good question. And we always try to define, okay, what's the next, if you could do this or that, would we solve everything? But it's very difficult to say, because it's also that you can't really jump ahead. You have to lay the full picture, the whole puzzle. You can't start somewhere else. You have to add it one to the other. So, for example, with the different biomarkers or clocks, we want to know, in order to get to know if they're useful in the clinic, we need to do all the validation studies. We need to do all of this responsiveness, and because for these markers to be validated as subclinical markers of aging, they need to be related to the hard endpoint, which is mortality or lifespan. And that is, of course, very difficult, because if we're going to study humans until they die, and we also measure the biomarkers and how they change, and then add on an intervention, etc., it's a huge study. It costs lots of funding, and it will take so many years to conduct. So I think that's why, of course, working with the aging and lifespan, it's super complicated, because it's like the last thing that happens in life, and then it's very difficult to do clinical studies on that endpoint.
Yeah.
But that's what we need, I think.
What about how these clocks generalize across populations and diseases, and is that a challenge as well?
It could be, but I would say, at least for the epigenetic clocks that are mostly we know most about those clocks because they have been replicated in so many different studies and populations, etc. We know that results are quite comparable across different settings and also across other like technical modalities, like how you normalize your data or how you modify your data in different ways. So it's quite stable with using, different clocks in that way. And normally they are trained in bigger general populations that are healthy, but that could be one thing, that there are also clocks that are trained for a specific purpose. So maybe you want a specific clock to be trained on cancer, then you need to have that type of population. There are also clocks for children or estimating gestational age and things like that. So that they're very specific to the context that they're also trained on. So we have to be aware that we have different settings, and then of course they are not generalizable across all of it. So it depends on what you're studying, but the overall general aging clocks like Horvath and Hannum, etc. I would say that they are fairly generalizable across different settings.
What about different species? Do you see the same there?
There are some differences, I would say. And ideally what you would do is that you would create and train a clock on the type of population that you want to study. So, for example, the Horvath clocks, I think it's mostly trained on European ancestry populations, humans from European ancestry backgrounds. So that means that it's not really transferable to another population, let's say Asian populations, etc. But now we have seen it's been done in so many different studies, and then you can again say that, okay, but the validation is okay because we've done it so many times also in other settings, so that's fine. But I would say that sometimes you also need to think about these things, that how you train different data, because if it's a relative measure as well, of course, then you will relate the epigenetic age of a population from Singapore, for example, to populations with European ancestry background that perhaps live in the US. So those are different settings, and then of course it's not exactly comparable if you use the same model and apply it to a different setting. So you always have to consider how much different is it, and is it valid to apply this to my data, to my context, or should I train a new model, for example? So it's not super easy to answer. It depends always, I would say.
It's biology.
Can I come back to a question I had earlier? You said we're looking at these as clocks or expressions that correlate with chronological age, and then, but not as causes of aging. But who is looking into methylation as cause of aging, as a causal mechanism? Or where do we stand there?
Yeah, you can do it in different ways. Of course, if you really want to understand cause and consequence, it's some sort of experimental validation that you need. So maybe you need to test that in different settings. So there are a couple of studies where it's been done in rats and mice, where they try to use to really change the epigenome marks, and then they have shown that by doing that experimentally, they can show that the epigenetic clocks in mice. So there are also these clocks that are trained and developed for mice. Then the clock values in the mice, they decrease, and they also show improvements in clinical symptoms. So for example, I think there was one study where they studied the retina of the eye, and they could show that there was improvement there. So you kind of need to show the whole scale, but I don't think that they had any data on like the hard endpoint on, okay, but did it also mean that the mice lived longer? I haven't seen that. So there are starting to be some of those studies. And in humans, of course, this is very difficult to do. There aren't any studies on that. You can use other types of methods. So in my work, we use a lot of statistical methods, and we also use genetics. So if you combine genetic data with epigenetic data, you can draw some conclusions on that, etc. And we have also estimated the heritability of epigenetic clocks, for example. So we know that they should be driven about 50% from genes. So in that sense, there is some sort of causality in it, because if genes are involved, it's really a strong factor for that. But you probably need to do a lot of different studies and look at the same question from many different angles with different methods to get closer to that answer, I would say.
Have we made any studies about how good we are at visually assessing people's biological age? How good are we to see whether somebody's aging fast or slowly?
You mean just looking in their face then?
Yes. So if someone looks at someone who's 50 and say, well, looks like 45 or 40 or something like that.
Yeah.
How well does that match up with the methylation test?
Yeah. I don't think that there is any studies that compare that estimate with methylation, but there are studies where they looked at basically people had to look at photos of individuals and then they guessed their age based on the photos. And just based on that, you can also build a clock, like a face aging clock. And that turned out to be also a good predictor of your future health. And now there are more AI tools, I would say, so machine learning tools that just looks at, or they basically screen images of faces, and then from that they build assessments. So you can make predictive clocks of facial aging, etc. It works like any other type of aging clock, but I can't say how accurate they are, and I haven't seen any studies where they've done these type of facial aging clocks and compare to methylation. We did, however, do a study in UK Biobank, actually, in my group, where we looked at, they have some sort of facial aging parameter there, and we could see that there was an association with, if you were considered to be older in your face, you also had shorter telomeres. So telomere length is also a kind of a biomarker of cellular aging. So we could see that on a population-based level. Of course, there would be a very large individual variability here, but there is some link probably. Mm.
So now, Patrick, when you see someone that you feel is maybe older than their age, you immediately think, oh, they must have short telomeres.
Absolutely, and now since I'm confusing causation and correlation, I'm going to apply a lot of skin creams, and I will live a long time.
Yeah, exactly. Yeah.
Yeah, that's always a hard one.
Yeah.
What I was going to ask you is do we know if these clocks, do they reach every hallmark of aging? Do we know, is there any hallmark of aging that we have a hard time associating with clocks?
Yeah, I don't think that we have a clock for all the hallmarks. And the hallmarks, they are constantly growing as well.
Yeah, I think there are 14 now.
Yeah, exactly. So epigenetic alteration, for example, that's just one hallmark. Telomeres is another hallmark.
Yeah, but I guess you could say that epigenetic alterations play a role for several other hallmarks as well. Mm. So I don't know if I'm phrasing the question right, but just is there any hallmark that we don't know how to build a clock around, if you will? Well, we actually just wrote a proposal for a grant funding where we want to build better clocks for autophagy. So that, okay, I think is one thing that they spelled out as an individual hallmark now in the new version of the hallmarks. Yeah. So that, for example, we don't have some other things you could measure experimentally. Maybe someone has had to set up an assay in their lab. But taking that and to measure it large scale in human populations, which is what I'm mostly doing, there may be a big gap in between that, so that you have a laboratory method, but it's not scalable yet for
for larger investigations. So
and autophagy has been hard to measure at all, right? It's kind of hard to know what's the best way to, if you're going to do a fast, when does autophagy kick in? You don't really know. It's just like an approximation of how that might play out, right?
Yeah, so what you normally do is that you try to look at gene expression or some proteins that you know are related to the autophagy pathway, for example. So of course you can do that type of thing for many pathways. Inflammation is another hallmark. So if you look at proteins that are related to inflammation, then you pick up maybe some of that chronic inflammatory pathway. But
right, from a proteomic clock, you could maybe say something about inflammation, and then you touch on another hallmark, right?
Yeah, exactly.
I saw some study about some kind of MRI-based clock. Do you know about that study?
Well, there are several now.
There are several. Well, yeah. So you're not using like a blood test. You're using imaging.
Exactly.
How does that work, and why is that interesting?
So I think, the brain, we know that normal brain aging is also something that happens when we age across the life course. And if you scan, there are different methods for scanning, MRI or PET and CT, etc. You can pick up some of the physiological changes that happens to our brain. So, the gray matter or white matter intensity and things like that. Which are, it's just a normal consequence of aging. They are changing, so they're shrinking or they're making, they're just changing conformation or something. Of course, there are also lots of pathological things that is changing in the brain if you, for example, have dementia, etc. But just trying to study normal aging, that is what I think these new studies are doing. So they're using different brains and images as scans, and then using machine learning methods to try to train models where you can pick up these changes that happens in the brain structure and relate that to aging as well. And based on that, you can build different brain aging clocks.
Yeah. And what about you? We've talked a little bit about proteomic clocks, but you also mentioned metabolomic clocks.
Yeah.
Also a step removed from epigenetics.
Absolutely, it's even further because metabolites are
right.
Quite big molecules. They can be very big.
Yeah.
And when you, depending on how, there are many different methods for measuring metabolomics, and depending on if you use what panel you use, you can pick up different, maybe you have the small ones or the big ones, and there's also a mix of, because a lot of metabolites you have circulating in the body because they were from external sources. So if you eat something, if you smoke, for example, you will have a metabolite circulating in the body based on things from that we are exposed to. But we will also have metabolites in the body based on normal metabolism and processes that are happening in our body. So we can also secrete metabolites from many of these cell cycles and other pathways. So it's a combination of both external and internal metabolic molecules that we can assess. And many of them are related to aging as well. So you can build the same principle again. You can measure different metabolites in your body, and then you can train a clock to predict age using the values of these metabolites and use that information to look at health risks. And that works very well as well.
Are these expensive, these types of different clocks?
I mean, there are some attempts now for trying to assess if they work in a clinical setting. I know that there are ongoing studies in Finland and in the Netherlands using metabolic clocks. But it depends on what you measure, I would say. I mean, normally all of these things, it's not something that is today affordable to do in the general healthcare sector. So they're not that cheap. But metabolic, some of the metabolic panels that you can use, some sort of assay, are cheaper than other kind of omics techniques like proteomics or the DNA methylation arrays, etc. So metabolites are perhaps a little bit cheaper to assess, I would say.
Have you measured your own biological age?
No, I haven't.
Patrick?
No, but I'd like to enter the competition. You know, Brian Johnson is only 36.
Oh, yeah. He's very young.
Chronologically 44, but he's in reality 36.
But again, it depends on what measurement he was using for that. You know, he has all sorts of different assessments depending on what he look at.
Do you know which tests he's using?
I think he's using some of the epigenetic clock tests, for sure.
I think he's using it's called Dunedin Pace.
That's one of the epigenetic clocks.
Yeah.
But there are several.
Do you like this competition? Is it good? Is the popularization good, or?
I mean, in general, of course, it's good that the topic is popular because it helps raise awareness and the public becomes aware and are interested in the type of research that we're doing. And for me, as a researcher, this is, of course, something good. I mean, to get the interest going, that's where it starts to get the really big improvement in terms of research funding, etc., and things, how you view it. But then, so I think in general, it's a positive thing, absolutely. But then, of course, there's always these bold statements and claims of things that, you know, we've solved it and everyone's going to be 200 years or whatever. That, from my perspective, it's not true at all. And that, of course, creates some sort of mismatch between reality and perception of the public says, oh, but this, you know, Putin and Xi Jinping discusses how they will live to 150. And to me, this is not really reality. We're not there yet.
Yeah, I think I lost some of my faith in direct-to-consumer epigenetic tests when I wrote my book, and I think I changed every biomarker there was. I lost a lot of weight and everything looked better, and then it turned out that I actually biologically aged a few years during that year when I turned around my lifestyle. So and I mean that we know that to be true. You can do these types of tests, and you probably, if you want to get some measure of reality there, you have to do several and maybe look at the average or something like that, right? And that could be quite expensive to do.
Exactly, and also the error margin of the best epigenetic clocks is somewhere between 1 and 2 years. So if you changed your lifestyle from 1 year to the next, that window is not long enough to counteract these, you know, normal fluctuations of just the assessment. So this is what I mean.
But you see these people, they're talking about, 3 months ago, my test came back and I was this age, and look, now I'm 5 years younger. Yeah, so that's a problem. But I guess I think, Sara, that you have gotten to do quite a few interviews, right? Because of this interest in that maybe people like Brian Johnson brings to the field. So it's a double-edged sword there. Absolutely.
No, but that's what I mean. In general, it's very good with the interest, but for me as a scientist, then it's also my role to explain where we are and where I think that no, these bold statements is not true, and trying to explain from my perspective where I think we stand today.
Okay, so we've talked about MRI clocks, we talked about metabolomic proteomic, epigenetic, probably some other clocks, PC clocks. Are there any other types of clocks that we should know about?
Well, absolutely. I mean, what we do in research is that we also build clocks based on other domains of data. So now we only talked about all the things, the omics that you can get from a blood sample or another tissue type, saliva or skin or something.
Yeah.
But you can actually look at other things. You can look at your function in the body. So if you do lung function testing, spirometry, you can look at aerobic capacity. If you do these, challenging tests, for example, you can also, in the similar manner, you can create a clock based on what is expected of your, if you are 30 years old, what is expected that your lung function should be. Because already when we are 25, 30 years of age, this is when the lung starts to, as a normal process, normal aging starts already then, and we can see how it actually goes down. And there are, of course, different, so this is something I always talk about as well, that you should be aware of what you want to measure at what time point in life, because different things change at different time points. So these omics clocks, epigenetics, etc., you can measure them across your whole lifespan. But then certain functional assessments can be useful on top of that, but only when they start to differentiate, basically. And like, at midlife, you start to have reading glasses. Maybe later in life, most people would actually need hearing aids, etc. So there are normal processes of functional decline with aging. And also muscles and the muscle strength and balance and these things are things you can quantify, and they can tell us quite much about the function of the body as well.
But you can clock children as well, right?
Well, I mean, if you talk about the omics-based clocks, absolutely. But you need to build them. There are clocks from epigenetic data that are built in samples from children. So then it works. Because also in some way, aging is just a natural continuation of the developmental and growth process that we see in children and adolescents. And after that, when we are grown-ups, it's just something that is continued, and then we call it aging, basically. We don't grow anymore, but it's a lot of the same cellular turnover things that is happening. And then, of course, in the old ages, if we talk about biological aging and function, etc., then it's much more relevant to talk about, frailty, if you're pre-frail, if you're having a disease, maybe you have several diseases, multimorbidity, sarcopenia, etc. Things like that becomes much more important for your health once you reach the really old ages.
If anyone is wondering why I stopped looking at you guys, I just realized that I think I wrote something about, I think it's David Sinclair's lab. I'm just trying to find that now. Yeah, there was a press release. It's a company called Biostate AI, and they claim to have created an AI scientist that can work 24/7, accelerating discovery. But then they did an experiment together with David Sinclair's lab. And so I can just read what I wrote. Biostate and Sinclair's lab turned Cadence, that's the AI scientist, I guess, onto one of the most critical instruments in aging research, the biological age clock. Cadence uncovered evidence that different sets of genes dominate at different life stages. The RNA transcripts that signal biological age in a 30-year-old are not the same ones that matter in a 70-year-old. In other words, aging appears to move through shifting biological phases, with molecular priorities changing as we grow older. Equally important, the clock produced by Cadence does something its predecessors never did. It reports not only an age estimate, but also how certain it is about that estimate. The researchers show that uncertainty spikes precisely at moments when biology itself is in flux: adolescence, midlife transitions, and extreme old age. That means the clock doesn't just measure age, it also highlights the turbulent passages when human biology is least stable. Did you know about this?
No, I didn't. I haven't heard that. So interesting. That
What? Say something more than that it's interesting.
Well, I have to read it then. But
Yeah.
I mean, it makes sense, of course, that because I mean, we talked a little bit about that the reliability of the clocks are not that great, and we try to measure that looking at technical replicates, for example, and how valid are results, and comparing. I said something that the error margin could be 1 to 2 years at best, etc. But of course, it makes sense that shifts along the lifespan, and of course in children and adolescents, when they grow a lot, there is a big difference, because we all know as teenagers, people grow in different and develop in puberty at different ages, and it's a huge variability there, of course. And the same in midlife. There is now a lot of, it's also a growing interest in looking into female menopausal transition, because that's also considered like a natural study of accelerated aging during that phase, because we've seen in now, if you look at clinical biomarkers, for example, during the menopausal transition in women, there's a huge difference. So really, in a very short time, you basically change your whole profile because of this transition. So it makes sense that this is also true. And in old age groups, we know that predictions are not accurate, because if you are 80 or 90 years old, it's very difficult to use these type of clocks to predict how long you're going to live, because a 90-year-old has maybe 50% chance to live another few years. I can't tell you the numbers exactly, but it's very, very difficult to say how long a person will live when they're very old, because there's so much else going on, or it's just randomness at that age. So it makes sense, absolutely. And that's a good idea to have those assessments of how accurate the model is at different ages as well.
I looked into it recently, and it said that when you're 90, you have 1 chance in 6 of dying, or the risk of dying is 1 in 6 over the next 12 months. So it's exactly like throwing a dice.
Absolutely. And we know in adults, every 8 years, you double your risk of dying. So it's an exponential growth there until the old ages, when it's very difficult to predict anything.
When you mentioned the menopausal stage, I got thinking about reproductive health. Is there an ovary clock as well?
Yes, there is actually.
Because I think that might be the fastest aging organ in the human body, right? And we haven't studied that nearly as much as we should be doing, considering that it ages so fast. So where are we on that now?
That's true, and there are groups now looking into that. So there is exactly this ovary clock, but there are also other organs. So it's been a lot of discussion about the thymus, for example.
Right. That's also a fast aging organ.
Yeah, or it really shrinks into a fatty organ, infiltrated with fatty acids, and then basically shrinks into nothing before midlife. So also we don't know how important that is for aging, but there are papers now looking at thymus aging clocks, and that's also related to health, etc. So there are absolutely different organs and systems that age prematurely, if you put it like that we haven't really studied yet in that perspective. So yeah, definitely we should do that.
I wanted to bring up our naked mole rat to say if they've tested clocks on that and what you get from that.
Yeah, so that's the comparative biology in aging research is very interesting, where they try to study different long-lived animals and species, and then compare and see, okay, how what is the mechanism behind the fact that these animals can live this long, for example. So naked mole rat is one such model. And they basically live 10 times longer than other mice or other rats. And I also think it's interesting because I don't think they don't get cancer, for example. So they have somewhat other types of mechanisms. And very often it has to do with DNA repair, for example, or improved autophagy, or it's fundamental cellular maintenance programs that they usually have optimized for their condition in that organism. So there are more examples of the Greenland shark, the bowhead whale, bats are also very long-lived species in general, which are interesting. And also I have heard some talks from researchers studying axolots, which are a type of salamander. And there are studies now doing the epigenetic clock in axolots, which is also one of these long-lived species. And basically it's stable, it's flat. I mean, for humans, it ticks like like a clock. But in axolots, when they are grown, it's nothing, it's just flat. So they don't really know what they die from. All of a sudden they just die, but you can't measure it from the normal, epigenetic clocks, etc. So it's super fascinating.
That is very interesting, because we've had discussions with other guests about this, and Peter and I say, but why doesn't it live for hundreds of years, right? I mean, why would it stop if its chance of dying is that of a young animal? So, but methylation status would be the same then at 2 years and at 30 years for a naked mole rat.
Probably. I don't know if there are maybe there are methylation clocks in naked mole rats. I'm not sure. But that's my guess. So, or more or less stable. And there are also other species. So we have, for example, killifish or C. elegans. We even have the bear, where species or animals, they go into some sort of dormant stage. So like they go into hibernation or dour states or whatever it's called in that organism. And also it's been shown then that in those states they also don't age. They basically put everything on hold. And after that, when they're going back to the normal phase, they continue to age. So it's super fascinating. There must be then mechanisms in nature that regulates this in different ways. And we don't understand all of that. Yeah.
Please find that switch fast.
The problem is that very many things that we do find in, for example, C. elegans, it doesn't translate to humans. Even between mice and humans, translation is very difficult. So that's why we also need to do the studies in large populations, like what I'm doing. And we need translation, basically. We need all sorts of studies. So that's important.
So let me just get back to the David Sinclair thing here. So what they spent a great deal amount talking about was this certainty or uncertainty factor of this type of clock. Is that important? Is clocks suffering from that now, that they can't show uncertainty?
Yes, I guess so. I mean, in research papers, we not always, but we try to add those type of measures as well. But I'm missing that type of assessment from clinics, because you can assess your biological age. There are clinics where you can do it, although it's not the public healthcare sector, it's private companies, etc. And I don't think that they really provide these numbers as well. And I think that would be helpful, because how would people know? Like you said, they're testing again after 3 months and they reduced their biological age by 5 years or something. But maybe it would have been helpful to also have the uncertainty level there, like how much error could it be in this assessment, and that could have of course, it's not good, I guess, for the company, the selling point to say that, oh, this is really not providing any valuable numbers, but somehow that's what we need. How are we going to improve it if we don't acknowledge the fact that they are not accurate enough today?
Yeah. And the other thing I was going to bring up is that this was made pretty fast. So David Sinclair had a quote where he said, "Cadence enabled us to complete an entire research study in just a few weeks, work that typically requires months or years of expert analysis. It pointed us to markers and pathways that warrant deeper study and helped us build a unified AI model for predicting biological age." So that brings us, of course, to artificial intelligence. I guess, I mean, the first Horvath clock was heavily based on computational biology and machine learning. But how is AI helping biologists like yourself now with all these new tools coming out?
No, I think it's a major change. I mean, in terms of how much data we can handle and the complexity of data that we can handle. So of course we need to apply these tools in different ways. So I think that it's mostly been different methods to train these type of aging clocks, and it's now been useful to also to combine different types of AI or machine learning methods, and then we can show that we can improve the clocks by combining them as well. So I think that's one aspect. And then, of course, in my everyday life as a researcher, I use a lot of these tools, and I'm sure you do in your work as well. So I think we have to make the best out of them, and we have to start to learn how to use them because they will be around, basically. I mean, now they're here, so we just have to adapt to that. And it will help us a lot.
But I was curious about your assessment of where we do stand in terms of solving aging, or to some extent delay aging or manipulate aging in a positive way.
It's difficult to say, when we're going to solve things, because very often what you hear is that, oh, but the first person that will live to 150 is born already, or to live to 200 years, etc. And we can never say, it's very difficult to predict what is around the corner, like in the future, what will we solve in the future, given these new methods and all of that data that we're producing. But today, I mean, the things that we know today in terms of intervention, supplements, drugs, lifestyle changes, etc., we will not solve aging by just the knowledge that we have today. So I think in order to live longer than maximum lifespan today, we need to solve some of the complexities of cellular aging. And how we can do that, DNA machinery and autophagy and those type of processes, epigenetic and cellular reprogramming. And I don't think that we're really close to do that today. There are experiments ongoing, but it's also something that is very, very complex. And one thing we forget when we talk about, the techniques and AI is moving so quickly, it's more about technology then. And here we talk medicine and biology, and that is something else. So I don't want to be pessimistic about it, but I don't think that we're anywhere close to say that the first person who will live to 150 is already born. I don't think so. But maybe Brian Johnson will beat us to it, but we won't know because we will not be alive then. So, yeah.
Which of the current paradigms do you find the most promising?
Yeah, I don't know if I can answer that, actually. I mean, it depends on what the question is. If the question is, okay, to extend the lifespan, then of course we need to solve some of the cellular things. But my research perspective is more that we want to understand how we can live healthier for longer. then of course it's much more important to look into preventive healthcare and how we can slow down biological aging by using maybe better estimates of biological aging, for example. So that is kind of a different question than, to get to the more extremes of extending longevity, etc. And there, to solve that, again, we need much more promising experiments, I think, in order to. I mean, we're not even close to try something like that on humans yet. So,
I was thinking you would be in a good position to evaluate epigenetic reprogramming, for example.
Yeah, but it's very, very difficult. I mean, we aren't close to, even if there are some papers that I said, it's also from David Sinclair's lab. And he sometimes claimed that, there will be a clinical trial now. He has said that a couple of times, but I haven't seen it. The problem with epigenetic reprogramming is that the whole machinery and the mechanism around DNA methylation and these methyl groups, the switch on different genes, there is an enzyme that is handling this in the DNA methyltransferase, and it's sequence unspecific. So it just basically happens wherever the chromatid opens up and the DNA molecule is exposed to these type of enzymes. So it also seems to be some sort of, it's a response to something else. So maybe the transcription factors that are sequence specific. So there is also complex regulatory things happening between these different processes, epigenetics and transcriptomics, etc. And yeah, so it's very, very difficult, I would say. And I can't say that I know all of the basics because I don't. But the way I see it so far is that we haven't really solved anything there yet.
What do you think? I mean, we could probably safely say that exercise is the best intervention that we have to slow aging now. When do you think?
I don't know. I would say that fasting is a better.
Fasting?
Yeah.
Yeah, okay. Even for humans, you think?
Yeah.
Yeah. Okay. Fasting is okay for me. These both exercise and fasting are two very old, tried and true interventions, and it's still the best we've got. When do you think we have an intervention that's better than fasting or exercise?
When I think that we have that.
Yeah.
Yeah, it's very difficult to know.
You're a Swedish researcher, not an American one. Okay.
Yeah. Yeah.
Cautious.
So, like all researchers that don't know what to say, then you always say, it's more than 10 years. Then it's,
more studies are needed.
But I would be happy to be proven wrong, but because I know that it's very difficult to see what's coming in the future and things are moving so quickly. But this is where we are today.
But also this circles back to what we talked about in the beginning. You said that there were probably fewer than 100 people working on this problem in Sweden. What should we do to convince the decision makers in Sweden, politicians, that this is the most pressing problem that humanity faces, and we have to really scale up our efforts now, because if we do, we can save a lot of lives. What can we do to make it happen faster?
Well, I think you already do that. You write popular science books and you're out talking and
I'm unsuccessful. I haven't moved the needle.
And you're having this pod and I mean I also try with we do a lot of public outreach and that's important and I try to prioritize that, but I mean, it's always a matter of fighting for the resources, and right now it seems to be more more resources going into the defense industry rather than science. So yeah, we're not really improving there at the moment.
What about the students? Are they interested in the field?
Yes, absolutely. So I also see a big change there, that there are a lot more students that contact me directly, and they really want to work on aging, and they know the work that I've been doing. And so it's also, of course, a matter of where you are in, like for me, where I am in my career, that now people know who I am and what I've been working on, which was different 10 years ago, obviously. But I think that there is a growing interest in students, and not just students. If you talk to general people, younger generations from the general, city in Sweden, many of them, they actually know who Brian Johnson is, so they've heard of different longevity influencers or different treatments, etc. So there is a general interest, and there are also these, the different longevity clusters and subgroups starting up at different universities in Lund and Uppsala and Stockholm, etc. So absolutely, it's a growing interest, and that's also a good start, because these people will grow up and they will continue to work on this, I think.
If you had $100 million to spend on your research, how would you spend it?
I would do a new data collection, I think, on female aging. I think that's super interesting, and also in together with some clinics where they do a lot of work for female health, to also set up data collections, but also maybe design a new clinical trial. We don't know exactly what happens in women when they go through this menopausal transition, what type of treatment is the best, and what treatment is the best for that person, because there is also individual precision here that you need to get to. And we don't know, for example, how do you change your biological age in these phases, and what type of treatment could mitigate this. So there's so much to do there. I would really like to do that.
Sounds like would have been great, and I guess that we do have a bottleneck in aging biology, which is data. We don't have enough of it. And with more data, we could pave the way for even better results using artificial intelligence, I guess. Patrick, do you think that do you have any additional questions before we ask Sara to recommend books?
Well, the question would be, Sara, have we forgot to ask you something?
I can't remember. No, I think we have talked about a lot of things. You really had a nice overview as well of the topic, I think. So you really know what to talk about already before I started talking. So that's very nice.
Yeah, we have our cheat sheet. Okay, Sara. We always ask our guests to recommend 3 books. Could be about anything. Did you think of any books?
Absolutely, I did. I really like that question, because that question I never answered before. So
that's lovely.
So then I have to look at my little notes here. But yes, so what I do, I like to read novels, and in my spare time, and I read a lot of novels in Swedish, because I work so much in English. So I want to have that in Swedish, and also a lot of women authors. But I selected 3 books that have maybe the common thread is, you can say aging, or you can say death and life trajectories, which I think all of that is relevant to the topic that we talked about today. But it's really novels and not, it's not popular science or anything like that. So the first one is called Unquiet, by Linn Ullmann. So that's a book. So Linn Ullmann, she's a Norwegian woman. So she's the daughter of Liv Ullmann and Ingmar Bergman. So Ingmar Bergman is the famous Swedish director. He also did a lot of movies about death, actually. That was one of his fascinations, I think.
Yeah. Immediately got to think of Persona. Which is a fantastic film. One of the best I've seen.
So I think he's well known, and this book was written about 10 years ago by his daughter then, and she actually describes Ingmar Bergman, and it's all about the different lives and things, but there are lots of sections in the book about his old life. So they have a lot of interviews. So there is this thread across the book about interviews between Lynn and her father, Ingmar Bergman, and you can really see how he has aged. Like he's a really old man here, and in the conversations that they have, it's fascinating. And they're doing all of this from his house in Gotland and the island Fårö outside of Gotland. And I've actually been there, so I did some research there because it's now owned by a foundation that allows scientists to come and work there. So there's all of these circular.
That's right. I was there. I wasn't in his house, but I was close by a few years ago.
But we were actually allowed to sit in the house and work as well. And we went to his movie theater that he has built there as well.
Yeah, and they do have some tours, but we couldn't find an available spot, so we had to settle to look, just glance at the house from the outside. Yeah,
right. But so this book is also all about this house and everything. It's it's really nice. I like it. Although I mean, he wasn't perhaps the most likable person, but
he would have been cancelled, probably.
Yeah. You know, we had 9 kids with 5 different women to start with. But anyway, it's a really nice book. I like it a lot. The second book is this Fresh Water for Flowers by Valérie Perrin. So that's a French author. And it's also written a couple of years ago. But I think this is very famous. It's been translated to lots of languages, and it's been very well discussed as well. But it's all about a woman called Violet, and she's a caretaker at the cemetery in a small town in France. So it's all about the cemetery and her earlier life. And then, of course, a lot about the death and people that comes to the cemetery and the people that works there and her life before that. And it's a fantastic book. I really, really liked it. So that's also, I mean, maybe on the topic of death, but so much more than that as well. And my last book is The Year of Magical Thinking by Joan Didion.
Wow, so that's wonderful.
The American writer. Have you read it?
Yeah, and also Blue Nights, the follow-up.
Yeah, exactly. Yeah, I read that too. So that's about the year that she was mourning her husband that died. And then her daughter was also very sick. It's really, a strong book, I would say, on that morning, and also, the earlier life that they had together. Basically. So
yes, she writes really, like no one else. Really, her own language.
Yeah.
Fantastic prose.
Yes. So it's also an old book. I mean, she's dead now, but I can highly recommend that. So those were my books. Not so much science, but
that's lovely. I kind of prefer that, because then I get to be, if we get someone saying David Sinclair, not that it's wrong to say David Sinclair's book. We appreciate everyone's recommendations, but adds another dimension when we get Joan Didion as a recommendation.
Yeah. And I would have liked to say your book as well, Peter, because whenever I'm out and talking in public about the hallmarks of aging and all of that, it's super good to be able to refer to a book in Swedish that goes deep into these hallmarks, and also discuss it in Swedish, and also you translated the names into Swedish. So I really appreciate that. I use that a lot. So just,
Thank you. Thank you so
we're
much.
all doing our work together
Yeah.
to lobby for this field.
Yeah. Now we're trying to bring that knowledge to the world. But I guess I have some competition there, but the more of us there is, the better, I think.
Yeah. Absolutely, but it's important to be able to speak to the layman in Swedish as well. Like it's that you have the knowledge and also the capacity to explain in your native language to the native speakers.
Yeah, and we also have Patrick's book, which is called Mot döden in Swedish, and the case against death. The philosophical argument for why life is good and death is bad. So it's a good combo, I think.
Absolutely. And then we have the more science lobbying book by Linus as well.
Yeah, we had him as a guest in episode 3. And now we're up to, I don't know, this is episode 34, I think.
Wow, that's fantastic. You're doing a good job.
Yeah, but you are doing a lot more important job, Sara. It's extremely important that we find the perfect biomarker of aging, and you are on a mission to help with that. So thank you so much for joining us today.
Thank you. I'm doing my best.